you can actually see your data set. So we can go to full study details. So again, these are the top 1,000. There are no studies. Regulated genes and biogroups, that's what I want. So here's a list of all our genes that we put in here. You can see nuclear receptor family had the largest fold change. You can click on here to see what it did. Here's the fold change. It went up 41 times. Now if I wanted more information on that, I could actually search by this gene. And if we highlight over here, you'll get a little bit of information. You can get whether it has variable regions associated with it and the proteins that it expresses. Ooh, I just clicked on that. So if you click on the protein, it brings you to this page, and you can kind of figure out what's going on here. Let's see. Obviously, this was our top gene in stem cells. I bet there's some stem cell stuff in here somewhere. Just gives you ideas on the, on the gene, what it does. All right, let me go back here. Yeah. You cannot have, the thing is you can have two things labeled the same, two columns labeled the same. So I think that's what happens. So either pick, and usually it's with the identifier, so pick one or the other. It should recognize AFI too. So if you pick the probe set ID, it should work fine. Okay, let me go back to my... Hold on a sec. Okay. So now I've selected my gene list. Now what I can do is I can find out. So I'm starting on quick view here. I selected my gene list. Here are all the genes in my gene list. These are basically the columns I put in. You've got fold change, we got p-values, all that information. Now here's what I can do is I can go to Curated Studies if I click on this. So these, basically what it's going to do is compare my gene list to all the other studies it has in its database. What do you think is going to be similar based on what we derive this gene list? What kind of studies you think are going to come up? Embryonic stem cells expressing. Knockdown in embryonic stem cells. S-group-grown knockdown cells, embryonic stem cells. What it's doing is it's basically telling me what, if I had no idea what this gene list is, I could look at the studies that my gene list is more similar to, and I would say, this is really similar to this one. Maybe these two have something in common. Then here's what I could do is I could add this to my project. Like, the gene list that I put in is very similar to this embryonic stem J1 cells expressing these two shRNAs. I can add that to my project and basically look at that data set later. And I haven't figured out, I know you can do like Venn diagrams with this, so you can take your data set and compare it to another data set and see how many genes are in common, which is an awesome function, by the way. Okay, it's added. Sure. Let's see, what else? We can go to BodyAtlas. So this will tell me where a lot of these genes that are being expressed or what kind of system is it expressed in. Right now it's going by body systems here, so endocrine, immune system. I can do this by ranks. So here are some of the different cell types where my genes are expressed. Esophagus obviously is relevant. I wouldn't expect adrenal gland, but who knows? That's interesting. This can tell you on this right side whether it's positively or negatively correlated. So that if in this esophagus one, so these are differentiated cells, they're kind of the opposite because these were like embryonic or esophageal stem cells. So they're probably the opposite, which is kind of cool. It's showing it here. You can see what cell types some of these genes are expressed in. I bet a bunch of embryonic stem cells. Hematopoietic stem cells of peripheral blood. That obviously makes sense. B lymphocytes. That I'm not quite sure about. But we do it again by ranks. Keratinocyte. Again, these are all seems to be epithelial, more kind of developmental stages of cells. But you can see, you can not only do this for, and again, you can export any of this information. And so that would be a good thing if you generate a gene list to throw through here. And what you can do is you can tell people, hey, all these genes, this is where most of the gene expression is known to occur. And if they're working on, say, adipose tissue and you pull up a bunch of fat, that's a good thing. Like here, I don't know why we're pulling up B lymphocytes. These are immature, so maybe these share stem cell properties of our cells, which is probably what's happening. What else? We can do pathway enrichment.